Hello, my name is Sara, and today I'll be presenting a recent study that had explored the effects of PDL1 and CD47 with in combination with OXP and FOLFOX chemotherapeutics in colorectal cancer models. Shooting at my screen now. Let's go over our table of contents. So I'll be going over the an introduction, a hypothesis, an outline of the study, key experiments, discussion, and a conclusion, as well as my references that I used throughout the presentation. So here, let's begin with an overview. So colorectal cancer, or CRC, is one of the most common cancers worldwide. It typically arises from polyps in the colon or rectum and can develop into malignant tumors, causing cancer. So there are common therapeutics used to treat CRC. There's OXP, and then there's FOLFOX, and then 5-FU. And OXP and FOLFOX tend to upregulate CD47 and PDL1, which are cell surface proteins known to evade the immune system in cancer cells. They target different parts of the immune system, both innate and adaptive. So CD47 acts as a don't eat me signal, while PDL1 acts as a don't find me signal. Just excuse my coughing, I'm so sorry. When treating cancer, therapeutics can induce immunologic cell death, or ICD, providing an immune response to combat these cancer cells. FOL, and OXP are inducers of ICD, and they do cause an upregulation of these immunosuppressive proteins. And this results in poor prognosis in patients, especially with MSS, CRC. And for some quick background information, MSS and MSI are biomarkers of which express the stability of DNA in tumors. So MSS is in the name, it's stable. The DNA within the tumor is stable. And MSI, which is also in the name, it's unstable. It there's likely to be mutations within that DNA, making it more responsive to CRC chemotherapeutics since it's more prone to mutations. So this study investigates how combining standard chemotherapeutics like OXP and FOLFOX with immune checkpoint inhibitors can improve treatment outcome in CRC and combat the upregulation of immunosuppressive cell proteins. So this is just to show you how CRC can look in the colon/rectum compared to how a healthy colon looks like. There's a clear visual difference with the hyperproliferation of cells and inflammation. It looks quite disgusting and honestly really painful. This is why studies and research like the one I'm presenting today are extremely important in helping to prevent such a horrendous cancer. So let's go on with the rationale/hypothesis of the study. The rationale behind the study lies in understanding how chemotherapy induces ICD. Um, the researchers hypothesized that dual blockage of PDL1 and CD47 in conjunction with these chemotherapeutics would enhance the immune activation, specifically promoting macrophage-mediated tumor engulfment and enhanced T cell response. The outline of the study. So the study used both in vitro and in vivo models. In vivo used CT26 tumor-bearing mice models, only females, 6 to 8 weeks old. In vitro just used CT26 cell lines. Um, so in vitro, there's CT26 colon cancer cells which were treated with OXP or FOLFOX to observe the expression of PDL1 and CD47. And the in vivo experiment, CT20, the female BLB/C mice were with the CT26 tumors were used to test how these treatments in combination with the anti-PDL1 and anti-CD47 affected tumor growth and as well as immune response. Lastly, the study went into detail about analyzing the immune cells in the spleen, drainage, lymph nodes, and then the tumor microenvironment. So there's some key experiments that I wanted to go over. Firstly is the in vitro expression analysis. The cells were treated with OXP or FOLFOX for 6 hours, and they showed a significant upregulation in PD-L1 and CD47 in their cell surface, as detected by the cytometry. Both OXP and FOLFOX regimen increases the expression of CD47 and PD-L1 on the CT26 cell surface, but FOLFOX increases the expression of CD47 and PD-L1 even more than OXP does. This confirms that chemotherapy alone activates immune checkpoint pathways, which could hinder the long-term efficacy unless targeted simultaneously. As you can see here, there's a large expression of the CD47 and as well as the PD-1 within the FOLFOX treatment in both the E and the F figures. As you can see here, also in the C and D, there's just a large amount of the expression. Um, So the second experiment that I wanted to talk about was the in vivo combination theory. Sorry, not theory, therapy. My bad. So the mice were treated with FOLFOX either alone or combined with anti-CD47 or anti-PD-L1 antibodies. And 3 days after the treatment, mice were euthanized and then cells were obtained from the tumor bulks. And then the FOL Fox regimen and the OXP significantly increased PD-L1 expression. And then furthermore, the FOL Fox increased both PD-L expression as well as CD47, but OXP failed to increase the CD47 in significantly. Combining both CD47 and anti-PD-L1 with either OXP or FOLFOX led to a significant increase in survival of the mice compared to both monotherapies with a chemotherapeutic agent and combined regimen. So overall, the results did show that combination therapy led to smaller tumors, better survival rates, and then also a stronger immune response, particularly in the CD8 plus T cells and M1 macrophages. The results do support the hypothesis that dual checkpoint blockage can enhance therapeutic benefits of chemotherapy, as we can see here in both the D and B figures, and using the key that with the FOL Fox, their treatment and the anti CD47 and PDL1 antibodies, we can see there is a large increase, a decrease within the tumor environment and the tumor itself. So let's go over the treatment details. So tumors were established by injecting 10 to the 6 CT26 cells subcutaneously, and treatments were initiated once tumors reached about 115 millimeters cubed, and the doses varied for specific treatments, different antibody treatments, but some dosages just were not included or like disclosed within the paper. Here's a dosage schedule of like the different days which different treatments were injected within the mice. So the results did support the author's hypothesis. Dual blockage of PD-L1 and CD47 significantly enhanced the therapeutic effects of FOL-FOX, but the study had limitations. It only used female mice, it lacked sample size disclosure, and it did not explore the long-term effects or toxicity that these treatments can provide. Some future directions I think the authors in study could take are that it can test across different CRC cell lines like MC38 and potentially exploring different kinds of dual blockage with additional exploring this specific dual blockage with additional immunotherapies like the 5FU chemotherapeutic agent. So overall, I think the study provides a really strong preclinical evidence that combining both FOLFOX and PD-L1 and CD47 inhibition can enhance anti-tumor immunity and provide better treatment outcomes in CRC. This approach does hold promising effects, especially for MSS patients who typically don't benefit from current treatments. But this may hopefully pave a new way for new clinical strategies. Here are my references that I used throughout the presentation. Thank you.